Okay, everybody, welcome back to the afternoon session. Great talk this morning from Jack. Now we're moving this afternoon with I would like to introduce Richard Beckett. Richard is an architect and lecturer of bioreceptive design at the Bartlett, University College London. He's the director of the RC7 on the B Pro Architectural Design Masters Programme and leads Studio 3 on the Landscape Architecture course. The researcher focuses on design operating at the intersection of computation, biofabrication and microbial ecologies in buildings and cities. His research on probiotic design won the River President's Research Award in 2021, but numerous projects has been exhibited internationally, including Archelab, Naturalizing Architecture, the Pompidou Centre and Nature Cooper, Hewitt Smithsonian Design Museum. Really looking forward to the talk, Richard. Richard's title of his talk is Probiotic Cities. Thanks, Richard.
Thanks very much, Darren. Yeah, of course, I'd like to start just by expressing my thanks for the invitation to talk today. I'm extremely grateful to Martin and Darren, who are the two people who've been super supportive in my work over the last few years, and Jack too actually has answered more than his fair share of my questions over phone and email. So yeah, I'm super grateful to you guys for sort of helping with the work I've been doing. So yeah, my name is Richard Beckett. I'm from the Bartlett UCL. So I'm an architect, so I'm a designer. So the talk today is going to be based on the design work that I've been doing. So of course I'm super interested in the science, in the microbiology, but I'm definitely not a microbiologist. But I'm a biodesigner, and so I've been working in the realm of design using living systems for quite a few years now. But the talk today is called Probiotic Cities, and underneath it, the term Probiotic Cities. It's very hard to say. So I'm going to talk a little bit about that and some of the work I've been doing, but I think it's important to give a background to how I became involved in this and really the underlying philosophies and ideas of why I'm pursuing this kind of work. So as an architect, when I first read a statement that design can play a role in shaping what the indoor microbiome is, that really kind of piqued my interest in this area, and this idea that yes, we have an indoor microbiome, we're surrounded by these microbes, and as Jack has already pointed out, it's been described as this kind of microbial wasteland, I think, and that really is because the way we design buildings and cities currently, we do everything we can to select for low biodiversity, for low microbiodiversity. Now, the advent of the microbiome and the kind of realization that microbes are not all pathogens, not all microbes are bad for us, raises this idea that we could also start to consider what might be beneficial indoor microbiomes, what might be a microbiome that may have more activity and could be beneficial for our health of humans, of buildings, and of course in the planet as well. So I'm using the word probiotic here in a very broad sense. It's not the probiotics of a yogurt that's maybe looks to put a few beneficial bacteria in your gut. I think I'm using probiotics as a much wider term and developing what I'm calling a probiotic design approach. And so the image you'll see here, I'm going to show you a bit later what this is, but probiotic approaches have been described by others as using life to manage life, and that's different from, or the opposite from, previous ways of controlling life, which have been described as antibiotic, and again a broader sense than purely the kind of the medical provision of an antibiotic. Usually our assumption of microbes as pathogens, as negative, has led us to these kind of modern ways of managing life that have been described as antibiotic. And so what you can see on the Petri dish there is a bad bug. So what's growing on the dish is MRSA, a superbug, that has antimicrobial resistance. And in the middle is a small disc of a probiotic ceramic material that we developed in one of our projects. And it contains a benign bacteria that comes from soil. It's a strain of Bacillus subtilis. And it can inhibit the growth of the Staphylococcus. And so what this is a method of managing life, but using life, okay, which is different from, as described, the sort of the modernist antibiotic approach. And if anybody's interested in what's been described as an emergence of a probiotic turn in the way that we manage life across a huge range of scales and environments. I do suggest that you might have a read of a recent book by Jamie Lorimer, which he called The Probiotic Planet. And he identifies this probiotic term across a range of scales, disciplines, and geographies. And most of these probiotic approaches are coming from, I guess, anxieties of knock-on effects of this modernist kind of antibiotic management of life that we've done. And so he identifies a series of probiotic mentalities across from landscapes to people that are looking to, I guess, reconfigure socio-ecological systems by using non-human agents. And I think, again, in the broader sense of what probiotic is, it really fits within the agenda of the Anthropocene. And it's people who are, whose goal is to do with multi-species flourishing and delivering human, environmental, and obviously planetary health. And so this is where we are. This is life where modern approaches to managing life have been predominant. And Jamie describes these as landscapes being ordered, cities and buildings have been sanitized, and bodies purified. And so this has brought a lot of benefits in doing this: economic growth, food surplus, disease eradication, and better lives. But what we are seeing now is evidence that perhaps some of these ways have gone too far. And Jamie uses the term antibiotic blowback. And I think what we're seeing is that for the first time, we're understanding that by always limiting microbiological and biological diversity, we're undermining many of our systems. And so at the landscape scale, we're seeing ecological systems have been undermined, leading to and driving species extinctions. In terms of agriculture, we've seen soil degradation, death of pollinators, and emergence of new diseases. Down at the scale of the body, we've seen the notion of dysbiosis, epidemics of absence, and the emergence of AMR, antimicrobial resistance. And of course, this is kind of sits somewhere in between these of what happens in the city too. Now, our cities or our urban biomes are probably one of the lowest cases of low biodiversity. And what we're seeing is notions of urban dysbiosis now. And this means our cities are, as ever they were, unhealthy places to live in, but they're also extremely low in terms of resilience to climate change and also to the outbreak of pandemics and the like. So when we look at disease trends, for example, we've always associated the presence of microbes with disease. Now, the emergence of antibiotics, and actually even before that, there were huge efforts to solve many of these infectious disease problems that came from microbes. What we did see, if you compare these two graphs, obviously the graph on the left is incidence of infectious disease from the 1950s to the turn of the century. But what we have seen is that while they've gone down, incidence of so-called immune disorders have gone up. And what this kind of shows along the wider consideration of scales of the planet is we're seeing these things what are being described as epidemics of absence. So these are new pathologies that are emerging, which come from our attempts to control life. And what we actually tend to be doing is creating even more challenging conditions for ourselves. And the COVID situation, as everybody's aware, has kind of brought microbes back to the forefront of our minds a little bit. And there is a risk that the modernist approach, the antibiotic approach of separation, sterilization, does risk pushing us further down this road. And as a kind of reminder that while we are focused on the moment about using methods to resist the spread and transmission of of COVID, that there is an underlying thing of AMR that still exists, which is a major threat to global security. And there is that risk that if we continue down this road of purely antibiotic modern approaches to managing life within our environments, there is a risk that we push these conditions even further. And so in the probiotic turn, people are starting to respond to these anxieties, and people are looking at different ways, new ways that not necessarily reject the antibiotic approach, but more consider about reconfiguring it. And so at landscape scales, we see people using ideas like rewilding, reintroducing species and keystone species into environments towards the aim of biome restoration. In farming, we're seeing a lot of organic approaches to permaculture and using living organisms for pest control, but also for bioremediation. And at the body too, the medical industry has embraced the idea that, you know, not all microbes are bad. The body needs a level of microbial diversity on it. And so, you know, exploring approaches such as fecal transfers. And of course, there's more people involved in post-Pasteuриan lifestyles. And of course, people already know pretty well about probiotic supplements. But the big question mark is to do with cities for us. And architecture and the way we design and build our cities has not, as it stands, shifted with this new kind of medical understanding of the microbiome. So when we were starting one of our first projects, we wrote a paper that suggested this. Architecture has a rich history in relation to health and medical understandings of health and well-being. And we made this argument that architecture currently is still in line with what we call the antibiotic turn. And we're making an argument that it's time for architecture to consider these new conceptions of microbiomes and health, and ask what future cities can be in terms of health and microbial diversity. And so if we take a very quick look at the sort of history of architecture in relation to health and illness, the picture on the left there is the industrialized city, where huge amounts of people were shifting from rural to urban living, and obviously came with that the dirt and the squalor and the disease that was rife in these places. And so what happened in the mid-19th century was something began called the Hygiene Revolution. And it was people started to realize that these conditions that people were living in were the cause of disease. And so a series of actors set about to modernize, sanitize, according to hygiene science, our cities and our buildings. Now, funnily enough, this wasn't led by architects. This was led by doctors, actually, and health reformers. It was actually quite a threat to the architectural profession at the moment, because architects just didn't have the knowledge of hygiene science in order to do this. And when we look at this kind of timeline of kind of architecture in relation to health, originally when this hygiene revolution started, miasma theory was still the sort of predominant understanding of disease, the idea that disease came from smells and odors emancipating from the ground. And so what we saw was a series of cleansing of the city, both physically, but also in terms of aesthetics, to protect, to deodorize from these smells, from this threat of miasmas. It wasn't until the turn of the century that germ theory really started to become proposed and perhaps more widely understood. But funnily enough, the majority of the hygiene changes based on miasma theory still held. The actual change in the understanding of disease as a microbial condition didn't actually have a major change on architecture. I think the biggest change actually came from when the discovery of antibiotics turned to more of their widespread use. And what really happened about that time was, firstly, that the diseases that had plagued the urban environments were suddenly solved. But it gave a sense of confidence over disease and infection. And probably what this led to was a shift in focus from designing cities from the perspective of health, probably driven much more by sustainability. So in its full flow, modernism was about exposure to light and air. It understood the sort of the beneficial roles that nature played in our health, while not necessarily understanding it from a microbiological perspective. So these two images here show an example of the open air school movement, this one in Paris. And the sanatorium movement really was architecture as a therapy. buildings were designed to be part of the treatment for tuberculosis, which in part was to do with heliotherapy, exposure to the sun, fresh air, and the notion of healthy and good environments. And I think modernism has always had that obsession with cleanliness, the idea that a good environment as the basis of health needs to be a clean environment. But remember, this is still all understood on the notion that microbes are bad. All microbes are bad. And so what we see is, from the kind of widespread use of antibiotics, as I've just alluded to, focus tended to shift, because I think antibiotics gave a sense of confidence that in many ways we'd solved disease, infectious disease, crowdsourced disease. And architecture kind of moved on to a much more climatic discourse. The event of technologies, here shown as kind of vacuum cleaners and sterilizing systems, but also HVAC systems, led to an architecture that became much more closed from the outside. So people were much more interested now in things like comfort, rather than necessarily exposure to environments. And when I was searching for, an image to put as the contemporary city, I couldn't really find one that fit with the agenda. But in many ways, the contemporary city is the majority of our buildings or cities are being driven by climate discourse and carbon conversation, so low energy buildings. The role of health, although is there, but many of our policies and approaches are favoured in terms of sustainability over health. And of course, the image on the right was a kind of fun thing that was going around on Instagram shortly after the sort of the COVID thing hit. But it really is our kind of default position that when there's something that's non-human and dangerous to us, our kind of first approach is to be antibiotic towards it. So the big question for me as an architect is to ask what would a post-antibiotic architecture be? And I think that really is in line with the notion of probiotic. And I think before we go on to show the work, I think there's an underlying philosophy that needs addressing to enable us to consider new ways for architecture. Because I think if we follow the science and the literature, there's a lot of practical steps of how we can improve biodiversity in our spaces. But I think if we fundamentally want to reconceptualize what our urban and built environments are, we need to understand a bit the kind of philosophy of why we do these things. And there's a sentence here that says there's an increasing consensus among scholars in various fields that the very idea of the human as ontologically different from all non-humans is precisely one of the main contemporary drivers of the planetary crisis. And I think that the conception of the human is one of the biggest shifts that the architecture needs to use in order to redefine what it's trying to do. And I think there's three contemporary shifts that raise fundamental questions about what it is to be human. Now, I think enough has been said about the microbiome from the scientific angle. And of course, if you read the headlines that, we're not as human as we thought we were, and the microbiome has shown this. But it asks fundamental questions of what it really means to be human. And it's a huge topic of which is beyond what we're talking about now. But I think if by taking the very concept of the microbiome, what we end up is with a contemporary version of the human that's very different from the modern human. And I think primarily the modern human was seen as an island, as a discrete body. And what that meant was, philosophically, man could be separated from non-man, from nature, from the non-human. The very presence of the microbiome, the fact that we are predominantly or a huge part of us is non-human, kind of suggests that separation is philosophically and physically impossible. And it means that any attempts that we do attempt to separate ourselves from those things are ultimately going to result in a negative condition. So the modern human is a biologically, culturally fixed category, non-relational as one, is replaced now by the contemporary human, dynamic and intermediate, ever-changing, relational, of course, with other systems and species, and of course the notion of the holobiont. Okay, we're not a single organism, we're actually a multitude. And I think the Anthropocene asked these similar questions. We've gone from being probably pre-modern, of unaware of our effect on the planet, unaware of the impact that we're having, to the contemporary human, which is fundamentally aware of that, which ultimately asks us to re-question again what we are. And the human is redistributed to the center of the Earth system, and with that dichotomy now that we have to act on non-human agencies as a fundamental part of our cells, which was previously out of reach. And I think the final point I'd make is understanding of diseases is shifting as well. So beyond just the idea that there are good microbes and bad microbes, I mean, I think that's super provocative for what architecture can be in terms of its microbiome. But what we also starting to see is that based on this model, disease is more than just microbes. So this idea that we had before, that disease as a unicausal relationship, means there's a microbe and it attacks a host. The microbe is an outsider and it attacks the human body and causes disease. But I think if we read Hinchcliffe in Pathological Lives, they're suggesting that disease is more than just microbes. And I think fundamentally what it means is people are really starting to understand the role that the environment plays, both physical and social, in creating health. And what they suggest is that life is always more or less pathogenic. It's the configuration of these microbe host environment spatial relations that either leads to a tipping point of causing disease or non-disease. So I think there are three fundamental shifts that architecture, if we really want to think of new ways to design our cities and buildings in terms of microbial health, we need to address those questions. And so just to finish before we go on to show some of the design work, a probiotic design approach then is definitely not rejecting the science and the understanding that we have of the hugely dangerous role that pathogens can play on havoc and the wider ecology and everything. But I think a probiotic design approach still looks to limit the presence of pathogens, but it also looks to permit the existence of other microbes that are not harmful, that are benign, that can potentially reduce the pathogenicity of spaces. And of course, it also then encourages those that deliver beneficial functions. And I think this is something that's looking ahead within the science of saying, when we hopefully may start to understand really what are good microbes for our health, what are beneficial for us, we could really start to consider what an architecture would be in order to promote those kinds of organisms. So I'm going to show now very quickly some of the student work we've been doing, because I think talking about beneficial microbiomes in buildings, I think initially this needs to start from the urban scale. And I think what we need to do is reconsider our urban environments based on the availability of microbial diversity. And I think parks and gardens and these open green spaces these are the main sources in urban environments of these microbes. We'd certainly like more of these, but it's not always feasible in urban environments. And so a probiotic approach might be more intelligent in that it starts to look at the connectivity between these spaces. And in the same way in London, as we have these kind of sight lines that operate so that people can maintain a view of St. Paul's Cathedral from various places in the city, we might start to consider wind movements as a way of effectively creating wind corridors for the movement of microbes around cities. And so we can certainly look at connecting diverse habitats on an urban scale. And of course, then we might be able to use wind simulations and the movement of microbes from these more diverse areas and start to plan our built forms around them. And whether that's identifying surfaces, roofs, walls, whatever, it would be a way of distributing microbes in a more biologically intelligent way around the city. Down at the scales of the buildings, we've done a lot of research into what we call bioreceptive materials. And so these were materials that were designed to support the growth of microorganisms, mainly cryptogams, so algae, lichens and mosses. And these were a bit of a contemporary consideration of the green wall, where the actual architectural substrate itself became promoted for biological growth. And I think these were often kind of proposed in opposition to, bare concrete walls that we see a lot on older buildings or infrastructure that don't allow any growth because of the physical and chemical conditions that's inherent in the materials. And so the idea that we might start making our buildings and cities out of materials that allow for this kind of micro-green photosynthetic growth is a fundamental part, I think, of what we might consider as probiotic cities. The big challenge, I think, that comes with this kind of approach, though, is that going back to what was shown in the previous slide, small walls happening on buildings, the odd building here and there, is not fundamentally going to change our city. And so what we do need is new ways to design based on these principles. So I'm just going to run, just press play, sorry, a short video here. And so one of our groups is looking at how machine learning can play a role in this, because I think one of the fundamental challenges to architects and urban designers is, how do you really understand the science and the large data sets that come from DNA sequencing and ecological monitoring? It's a hugely complex issue that can't be kind of narrowed down into sketches and things like that. And so we're exploring the idea that machine learning can help us with this. So what we can do is plug in ecological data, ecological understanding to do with environmental systems, orientations, slope angles, material bioreceptivity, into these GAN systems that we can then train data sets in order to define urban conditions or building conditions that are optimized for the growth of biodiversity. And here you see a group proposing quite a speculative looking architecture, but it's always driven by which way slopes are facing in terms of their orientation to the sun, levels of shade they get, but it's not happening on one single wall, it's inherent within the building. And here they are plugging in surrounding green information. And now that they're showing, using these data sets, how you can apply them to sites. So by taking kind of disused sites or perhaps post-industrial sites, we can feed in this environmental information in terms of large data sets, which probably exist as numbers, as Excel spreadsheets perhaps. And then we can use those to start configuring the building mass, but also its relationship with surrounding ecologies as a way to promote diversity. Okay, so here they are applying it to a site next to a railway track. And then as you see, as the data set starts to train itself, what we end up is with a series of building masses which are predominantly driven by non-human agendas. Okay, it's an architecture that of course is still considering as a design for human, but here we're using non-human agencies to lead that. So we see some final representations of a new kind of architecture. Okay, it's not one you might necessarily recognize in a city, but perhaps as we're looking for new ways, we might be more exploring in that. Sorry, it's a bit glitchy, the video, maybe it's a bit high. And just coming at the end of this video is actually a different group, but using the same process. And what you're going to see at the moment is obviously we can then use these same systems to design the floor plates of buildings. And what we're aiming for again is that we end up with a series of floor plates, of which you're going to see here. So here's a same process to devise a building mass that's determined by non-human agency. And what we have here is a series of floor plates that are purely driven by non-human agency. So these are not designed in the traditional sense of an architect. Instead, they are floor plates driven as a way to maximize non-human agencies within buildings. And I think what this is doing is, from a probiotic perspective, it's fundamentally challenging and questioning the role of the architect. I think the other main agenda of this work is that I think if we really want to achieve probiotic cities from the discussion as I've outlined, architects are going to need to work with people from other disciplines in the same way that we work fairly regularly with structural engineers. I think architects are going to need to work with ecologists, microbiologists, virologists. And the challenge is always the language, and the challenge is always the difference in design approaches that probably most of us here know very well. And I think these kinds of machine learning systems potentially offer a way to do that, where we can take data sets that have no visual representation, and we can start maybe plugging them in to really then create these rules and data sets for buildings that could optimize towards this probiotic condition. I'm going to skip through that one. So the project I'm going to show now is one of the first projects we did, which was actually in response to the spread of AMR. And there was a small amount of funding that came from the AHRC here as part of UKRI. And what we proposed to do was develop a probiotic way of limiting the spread of AMR in buildings. And so when you look at the diagrams that exist in the literature, this is the kind of the stuff you see. But of course, buildings are much more complex than that. I'm very grateful for Jack for already talking about the science behind most of this, so I won't really need to here. But what we started to do was in a very basic biological sense, was start to look around the building. And we didn't have a huge amount of money in this project, so we weren't doing any sequencing or metagenomics or anything like that. But we started very simply, and we chose a kitchen space for the reasons that have already been discussed. There's potentially some moisture there, there's some high level of use from different people using the kitchen space. And we just started to look using contact plates and just plates placed around the type of microbes we get there. And probably is not a surprise to anybody involved in microbiology here. There were a lot of pathogens present that probably came from the people using the kitchen. So these were taken and analysed in the lab. And of course, there was some MRSA there as well. And so kind of developing on and reading around the literature that exists, we started to look at the way the spaces were used, what kind of activities people were doing there. We were looking at the environmental conditions of temperature, humidity, to try to build up a picture of the space from, I guess, in a way, from a microbial perspective. You know, what could we learn by the way the spaces were used, how many people use it, how often people were coming in and out, what were the relationships between the building layout, but also things like the HVAC systems that we know is also a big kind of mover of microbes around buildings. And so this was a very simple basic level kind of attempt to do some kind of microbial survey on the space. But the overriding agenda was saying, could we develop a material that is living, containing benign microbes that could prevent the growth of the AMR pathogen, in this case Staph aureus. And so we worked from really the scale of the material upwards, starting with this idea of a material micro hybrid at the top. Could we create a material that we were able to get bacteria into? Could they survive then within that material long enough to effectively exhibit some kind of antimicrobial action? And then as we moved up in scales, we wanted to develop this into something that could be applied to a building wall, floor, as some kind of probiotic surface. And so initially what we did, we explored a range of materials. And this was all based on this idea of kind of bioreceptivity and materials that could be supportive for the survival of microbes. And so porosity, pH, surface roughness, these kind of physical characteristics of materials was really the driver. And we tried a lot of materials, from concrete, different types of concrete mixes, lots of ceramics. We tried aluminiums, rubbers. And what we did was produce a kind of a whole bunch of these materials that we could then take through this process of growing the bacillus to a set ODI in the lab, and then we could sterilize and then inoculate these strains into these materials. The porosity of the material allows the bacterial culture to be absorbed in. And then the idea that we would just leave them in a lab for a while untouched, and then we would replate them to effectively get a sense if the microbes were still alive in the material. And we saw pretty early on that certainly some materials were better than others. There were certainly some materials that the bacillus didn't seem to be able to survive in, certainly past a few days. And so this kind of just literally sticking these cubes back onto a growth plate and seeing if the organisms were still alive in there was really our kind of main approach at this time. And then we started looking at SEMs to see really, you know, where were the bugs in there, what did they look like? And, you know, what we clearly saw that definitely some materials were better than others. Certain levels of pore sizes were definitely playing a role. And I think really at this point, the material agenda wasn't specified. We were kind of fairly open to whichever material worked best. But I would say it was an agenda of the work that we didn't use sort of overly expensive materials or materials that couldn't really be applied at the architectural scale. And so materials like 3D printed aluminiums and stuff, perhaps some of the rubbers, although quite interesting, probably at an architectural scale just become too expensive to be viable to use. What we really found was that the concrete and the ceramic materials really seemed to be the best. We saw real good evidence of biofilm formation. And what we also saw was that certain material conditions change the sort of the properties of the bacteria while they were in the material. And as Jack, I think, has already made out in his talk, is that the lack of moisture in indoor environments usually makes them pretty inhospitable to microbes. Now, we wanted a system that would allow the microbes to survive within the material, but we also weren't particularly interested in complex irrigation systems where we're constantly feeding moisture and nutrients to these bugs to keep them alive. It was seen as a much more passive thing at this step. And what we did see from the Bacillus subtilis, in that certain materials, we saw that after a period of time, they were forming into spores. And I think what this does is offers a way of having a kind of an underlying probiotic material agency within a building that doesn't need constant maintenance. They can probably survive in there as spores for an indeterminate amount of time, and then can be reactivated potentially on some kind of signal if a microbe or a pathogen does appear. And so I think what we did eventually was we moved forward with one material that was doing very well, one of our ceramic materials. And so we set out to kind of quantify this. And so we developed a methodology using a very scientific method here of bashing and crushing up these discs, of which, oops, I'm just going to jump back, of which we could then just use kind of cell counting to see that. And so what we got from that was a kind of evidence that the bacillus could survive within the material, certainly for up to 30 days, but realistically probably much longer. And so we had this first idea of this material microbe hybrid. It was a living material. And so our next step was to prove or test that this could really inhibit the Staphylococcus. I'm sorry for the low-res graph on the right there. I'm not quite sure what's happened. But the very first thing we did, just to get a sense of this, was to do this kind of just literally putting this material onto a plate with some staph, and it gave a good suggestion that it can inhibit it. And so we went on again using a similar methodology as before of crushing up these discs using cell counting. And what we saw was that the limit of detection in the co-culture showed that the bacillus was definitely inhibiting the Staphylococcus. So this was, in its most basic inherent forms, a first example of a probiotic material. It's using life to manage life. It's using a benign strain of bacteria to prevent a harmful strain from growing. And this could, of course, then be considered to be applied within buildings. So what we did was look to scale up this material from kind of small cubes as a series of surface tiles. And I think probably one of the biggest interests to me as an architect is what really is the spatial configuration of microbes within buildings, and how would we then consider starting to design surfaces to deal with that complexity, that spatial complexity. And I think tiles, firstly, are a good way of doing that, because tiles are cheap and quick and easy to make, and they can be applied on surfaces because they're discrete elements. They could potentially be seeded with different kinds of inoculants. I think the bacillus as one is a good example, but I'm sure there are plenty of other beneficial or benign bacteria that may play important roles in limiting pathogenicity or even being healthy. And so the idea that we could apply these over different layers, and look, we applied this certainly to the kitchen as the idea of we could create a kind of backsplash area that would prevent AMR growing on this surface. But thinking about it more spatially in walls, we could also develop these systems where, we might have a series of tiles at low level, down at the sort of toddler level, that would have a very different microbial community within them than perhaps might have at the adult level. And again, based on the complexities of buildings, this could be something that could become spatially distributed with discrete elements. And then just building on that is that these materials also, as well as containing a probiotic organism within them, could also become a source for microbes within a building. So we heard today a lot about how people spread microbes around the building towards surfaces, but it does happen the other way as well. And so I think the idea that these surfaces may also shed materials or shed organisms into buildings that could be protective or potentially healthy is another interesting kind of fact. So we did a lot of work on, as well as considering just having the microbes in these tiles, where might we put them in the tiles, and how might we design these tiles in a way that could both catch pathogens perhaps, but also shed pathogens. And so we used a lot of computational approaches, CFD simulations, to understand how these kind of micro textures could firstly provide the habitat for the organism, but secondly shed them into other areas. And so this was now coming to the end of the project, and we fabricated a series of these tiles. It was four sets that could be then orientated differently to create variation in the spatial idea of microbial presence. And obviously, we're getting to the scale here where it becomes more and more difficult to work in the laboratory. So small discs are very easy to put into the autoclaves and sterilize. Tiles actually not too bad easy, but once we consider getting up to architectural scale pieces, the idea that these things are taken to laboratories and sterilized and done in a controlled manner becomes fundamentally challenging. And so we're also interested in other ways that we can do that. And so here, rather than having someone inoculating each of these tiles, we set up a robotic system just using a syringe pump and an XYZ positioning system of an alternator as a way of automating the inoculation stage of this. And so here we are with our final probiotic tiles. We were unable, unfortunately, in this project to put them into the kitchen and do any kind of testing on them. But we did exhibit the project as this kind of prototype probiotic kitchen. Just being aware of time, I've only got 2 more projects anyway to show. I think the next big challenge for me personally as an architect is to start to ask, well, if we can put living microbial agency into buildings, into materials in buildings, is it really going to have any effect on the microbiome of the space? And so we got a small amount of funding to do a very simple experiment into this. And so the focus switched to my office because we can always experiment in our own spaces, right? And so the first thing we did was start to look at the CFD of the airflow movement within the space, really as a way of locating the probiotic prototype. Where in the office is best to put this? This kind of addresses a bigger question, is if we really wanted to consider probiotic surfaces for buildings, just how much of our surfaces for buildings would need to be inoculated with bacteria? Is it something that would need to be everywhere? Or is it something that could be done in selected controlled areas? Really as a way of considering spreading these beneficial agency through the buildings and spaces. And so understanding how airflows work in buildings in relation to windows and HVAC systems could play a role in this, I think. And so what we did was use CFD to, firstly, was to identify where we would put our prototype, because we only have a small amount of money to make a few pieces. We can't cover the whole office. And so we did that here, and we identified that potentially this space was a good one because the HVAC system was blowing out and kind of causing turbulence on this wall, really. And then what we did was selected 10 points around the space that we would swab and send off for DNA sequencing. And we devised a sort of a before, during, and after approach. So what we did was we took 1 swab a week for 3 weeks before the prototype went in. So that was perhaps an indication of the existing condition. We then put the prototype in, which contained, I'll talk about it in a minute, but it contained a soil community, perhaps. We measured it for 3 weeks while the prototype was there, and then we measured for 3 weeks afterwards, once the probiotic components had been taken out. And we also had data loggers in there to monitor environment and things like that. So I'm just going to take you through the process. Now remember that we're interested in the role of design. So here we're using kind of CFD and wind simulations to drive where we might put the prototype, but also how we might consider the form, the geometry of the prototypes, all based on this idea of not only collecting microbes, but also shedding beneficial microbes into the environment. So we designed these series of components that were driven by these wind simulations, and using kind of 3D differential growth algorithms. What we ended up was a series of 3-dimensional components that could be fabricated using robotic extrusion. These were going to be made out of the clay ceramic material that we developed in the lab. And then, whoops, next one. And so here's the fabrication of these. And I think there was definitely an agenda here as an architect to move away from just tiles and flat surfaces into something that firstly is much more 3-dimensional. I think really pushing towards asking what architectural walls, spaces and furnitures might be. So here are the prototypes printed in clay. This was done with the guys from Sierra Lab. These were then bisque fired, which turns them into ceramic. And then what we chose to do was just to glaze the back sides. This added a level of non-porosity to the objects, but also gave them a much better structural strength. But what we did was leave the front sides open. So this is now our porous ceramic material, really envisaged as a kind of a wall component, but very three-dimensional. And these were inoculated with soil microbes, but here you see the difference between the glazed and the non-glazed areas. We also have these kind of long fingers with these kind of almost like cup conditions, which would allow air to circulate, and basically designed to really kind of maximize the air turbulence as a way of shedding the good bacteria out into the building. And so here they are on the wall, and then we undertook the study. Unfortunately, this study happened just as the COVID situation hit. So in terms of the DNA data, the sequencing data, we don't have any of that yet. It's definitely going to be happening. It's just where and when. What we do have is we put a lot of culture plates around and also spread some of these swabs onto culture plates. It would be very, very difficult to make any kind of conclusive statement at this point. But what we can definitely see changes in the types of organisms in the space, in and around the space, from when the probiotic installation was put in. And there definitely does seem to be some kind of spatial limit to how far these microbes are moving around the space. So we're definitely seeing a change in the microbial communities within about 1.5 meter radius around the piece. We're not necessarily seeing stuff happening on the wall on the other side, which is perhaps 2.5 meters away. So there's definitely some kind of spatial thing to consider here. And then, look, I'll finish with this one. It's probably coming to the end of the time. Again, the agenda of myself as an architect is to really fundamentally question what probiotic buildings the cities would be. And so scale is a constant issue to us. How do we really start to conceive these things? And so the project that's just started is the idea of creating a probiotic pavilion prototype. And this is a sort of a small scale prototype that could be indicative of what probiotic buildings might become. And really it's this two-sided pavilion that on the outside is using bioreceptive materials to grow mosses, algae, those kind of things, while on the inside it will be seeded with benign bacteria, probably bacillus, potentially some other soil microbial communities. And it really creates this sense of enclosure that one could enter and be exposed to these kinds of microbes. Currently just making the prototypes for these, so we're testing the fabrication method and a way of doing this at the large scale, trying these on flat panels and developing the materials. But this hopefully is going to be made within the next few months, and the plan is to put this into an environment. We're going to have 2 of these. One will be seeded with or inoculated with benign bacteria, the other will not. It will be a control. And the plan is to allow people to use this. Means we're going to allow people to sit on this, to touch this, to engage with it, and then we'll be using DNA sequencing to monitor what happens to the microbiome of the piece over time. So that is to come. So just to finish, of course, all this work happens in collaboration. So I'm hugely grateful to the people listed in here and the support we've had from BMAID and also from the AHRC from our funding. That's it.
Thanks, Richard. Get a clap. Well done. It was a really interesting talk. I mean, there's plenty of questions in the chat, so I think what we'll do is move over to have some of the questions as well.
Great.
So the first question we got is, do we know or have any other studies on how the growth on bioreceptive materials influence microbiome in the built environment? So are there any comparative studies?
I've not actually seen anything directly on that. I know there will be stuff coming out in the next probably year, because I know that people are starting to work on that now. I'm not sure I could point you to anything directly yet that's done that.
So there are no kind of groups or forums currently set up for this kind of work.
Yeah, it tends to be happening more in individual cases, I think.
Okay, and then the second question is: do these designs suit or support specific types of function of buildings? So
one more time, Darren. Sorry.
So when I think this was talked about earlier on with respect to the new design landscape of the buildings that you showed in one of your images earlier on. And you were saying, do these designs suit or support specific types of buildings with specific functions? Okay.
Yeah, I mean, it really is not considered from that specifically yet. I mean, again, we're being led a lot by the science, and obviously there's a huge amount of stuff going on in hospitals. It's going to be very difficult to do a lot of this stuff in hospitals, but certainly the home conditions and office spaces, I think as we kind of learn more from the sciences, it's definitely going to be potentially fitting those kinds of typologies.
So the third question was from me, and it's about, so you showed these kind of these green parts of the city where you could maybe improve the flow of microbes through a city. I mean, as we run those models, did you have any influence on that with height that you would need to? So, does this need to encase the top of a building, or is a patch of land okay?
Yeah, I mean, look again, when I read, I mean, I'm not an expert on this, but obviously you get variations in sort of biomass in terms of heights, right, up at different levels of aerial levels of microbiodiversity. I mean, the simulations we're using are not really from what might be used perhaps by engineers. I mean, these are much more design-led simulations as design tools. But I think, yeah, again, if there exists people that are studying this kind of stuff, so we could really get a better understanding of how these communities behave at different aerial heights, we could start to configure our cities based on that. And again, a lot of the work I'm talking about here is proposing fundamentally new approaches to building cities. Of course, there's a lot of stuff we can do that's much more pragmatic and much more to do with retrofitting already what we've got. But I do also see it as a role to explore the much more speculative angles of, if we want to really create new environments, what would we do? And I think in that sense, the idea that you could start planning building masses and typologies based on this environmental data, that for me would be a fundamentally different city from the one that exists now.
Yeah, I completely agree. That's great. So the next question is from Ben, and it's, do we need machine learning and large data sets to design buildings which can support complex ecosystems, or would it be better to understand the required habitats and conditions, so that architects working with ecologists could combine these requirements with other functional requirements for the building to thoughtfully design biodiverse buildings?
For sure. I mean, it definitely doesn't have to do that. I mean, the challenge has always been, I've seen, is how do you take the data that comes from something like sequencing and turn it directly into designable conditions? And at the moment, I find that a challenge, and that's just one aspect of an ecology, isn't it? And the thing we have learned from all of this science is how variable and how changeable these microbial conditions are at different times of the year, and different seasons, different times of the day, even how they're occupied differently. And I do have a worry that there's a complexity there that can't be fed into simple diagrams. It can't be fed into a ventilation strategy diagram that we're used to dealing with when we're dealing with one condition that's a fixed condition, sunlight coming in through windows. We can draw that as diagrams. Can you simply diagram microbial communities in buildings? I would guess probably not. And so the reason we were trying that is just to try and develop ways that we can deal with this kind of complexity. And I think there is a fundamental underlying thing that I kind of mentioned as well, in that as humans, we are human-centric. And even though many of us are interested in looking at new ways, fundamentally, we will always be human biased by our very nature. And so there is a potential, even though one might argue machine learning is still human, there is an argument that we might give up some of our human control through the use of these systems, where the data is trying as much as it can be to be led by non-human agency rather than human decision making.
I mean, I think the one note of regarding this from the suppose from microbiology side is that, I mean, is what is a pathogen, I think, is a big part of this, because I think a lot of the bacteria we see that can, cause infection and disease are usually opportunistic. And they tend to happily soil bacteria or carry around in as a percentage of your gut flora, or colonizing a part of your nose. It's only when they pick up these kind of pathogenic traits they become. So actually, we need to be careful. We're not trying to clear away species of bacteria that are still a part of that diverse community. Yeah. We need to. It's going to be quite a difficult challenge to kind of pick between that, but an interesting one to go forward. So one of the questions talks about, did you look at the how quickly these receptive materials degrade? How quickly?
In terms of the physical degradation,
I think so.
I mean, look, definitely using materials that are porous, and where they're going to be taking on water and going through environmental cycles, they definitely don't last as long in the same way as non-porous structural concrete materials. But again, I think it's a reconsideration of how we want to apply materials to buildings. These don't necessarily have to be the structural parts of buildings. They could be much more kind of cladding-based materials that are there for a set amount of time and then, need to be replaced or something like that. And certainly as an interior condition, I don't see why these materials wouldn't necessarily last as long as other materials. But aesthetics of growth and potentially mold and these things, it's a challenge for architecture, isn't it? Because we're so used to seeing clean, non-porous, flat materials.
Yeah, I was really interested to see that, at the back end of this, after you've kind of tested, that it's a clonal population still, did you do any other selective media plating to try to kind of stratify between if there was anything else in there? Because some things can tend to look like others.
Yeah, no, we didn't, to be honest, no. I mean, it was a very small study, this one, so.
No, it's still really, really good. And then the last question really is regarding the images of your office. And then somebody said, is that AI generated?
No, it's not. It's not AI generated. The idea was to, we know that the ways microbes move around buildings, from touch, from breathing through these HVAC systems, and so it was a way to start to engage with those kind of systems. So we were using the CFD of the airflow movement within the space, really as a way to locate the installation at that time, but also through this idea that, as well as just containing microbes that can potentially inhibit pathogens, could these things also become sources of microbes by spreading around the building? And I really like the idea that we might have thresholds for buildings that are extremely diverse, and it's as people come in and out of buildings that they pick up some of this kind of microbial diversity and then carry it almost through the building, rather than this idea of saying, well, we now suddenly have to start inoculating every single wall and desk in our buildings. I mean, it's not feasible, is it? So, by understanding how microbes move around a space, we might be able to design architectural geometries and conditions that promote that condition.
I mean, I really like the way that you've kind of already kind of worked out that about 1.5 meters away from it that you're seeing the signature, which is kind of, I suppose you could augment with fans blowing across it and all kinds of, but also in the air movement in your office. So I think there's so much nice work to still be done with it. So I think that's the end of the questions, Richard. So
Great.
Many thanks for another interesting talk, and I really enjoyed that. And thanks very much from the audience as well.
Thank you.
Cheers. Thank you.